Today we are going to play with some hydrochloric acid, but don't worry, it's dilute hydrochloric acid, so everything is pretty much safe. My dear children, we are going to do the mitosis experiment where we have to spot the stage of mitosis and we have to report that. Now, this experiment is something which is compulsory for all of you to do. There is no lottery system up over here, which means that we have to be pro at it. So, let's see what is this experiment all about and what are the requirements for this particular experiment. So, the first thing you would need onion root tip, because we are going to observe the stages of mitosis in the onion root tip. This is the stored one in your school. Things might be different. Please check with your teacher. Now, going ahead, you will require dilute hydrochloric acid. This is for softening up of your tissues out there, and yes, and it makes sure that the cells which are there, it takes up the stain. What is the stain we are going to use over here? We will be taking up acetocarmine stain over here. Now, you would also require watch glass, okay, and onto this watch glass what we are going to do is, we are going to take the onion root tips, we are going to put it over here, and then we will add dilute hydrochloric acid along with what? Acetocarmine. When we put dilute hydrochloric acid, it makes sure that the stain has been taken up. After that, we are going to heat it. How? With the use of spirit lamp. Now, again for holding it, we have a holder as well. Okay, we will just heat it for 10 to 15 seconds, that is all, not more than that, and just we will keep it aside and we will cover it as well so that it does not dry out. Post that, we are going to take that particular specimen and then we are going to put it on the actual glass slide where we will further do the experiment. Okay, I hope it makes sense, and you would also require the brush for the transferring purposes, and you will also require the blade as well. You will see why is it required in this process when you see this experiment. So, let's go ahead. So, firstly you need to take a few root tips onto a watch glass. I have taken only one, but you can take more, and then add few drops of acetocarmine stain. This is very important. And the same place itself you need to add few drops of HCl and heat it up with the help of a spirit lamp for 10 to 15 seconds. Then keep it aside for 30 minutes and make sure you cover it up with the help of a petri dish. Further, after 30 minutes, take this up onto a clean glass slide. This is the root tip that we need, so cut off the extra portion. Further, add few more drops of acetocarmine and then put the cover slip and dab it up with the help of a blotting paper so that it removes the extra stain, and also press it gently and then view it under the microscope at 10x power. And finally, we did this experiment as well, and my dear children, this is what you will get if at all you do it properly. Okay, so you would be able to observe multiple stages of mitosis, starting with the first stage, which is prophase. So prophase, this is a cell, and over here you can see that everything is clumped up in the center like this. So this means that the chromatin basically which is there, the chromatin fiber which is there, it has condensed and now it has formed the chromosomes. Apart from that, the nucleus membrane which is there, nuclear membrane which is there, it also starts breaking down at this particular point. So prophase, it looks something like this. Now metaphase is the next thing, where in the equatorial plane you would be able to see all the chromosomes are lying up in the middle. This is metaphase, M for middle, you can remember like that. Now in anaphase, they will start moving away from each other. Okay, in anaphase you can clearly see there is a movement on both the directions out there. Now telophase, it is kind of not very easy to spot, but just understand the chromosomes which are there, actually very easy if at all you get an understanding of it. So the chromosomes basically move completely towards two different poles. So they will be there towards two different poles. So anaphase looks like this, where they are still separating and they are moving away. You can clearly see they are moving away. Over here they have completed the movement, they are towards both of these ends. So this is what you can observe. Now as I always say, in your institution you might have had different steps, but there are certain steps which are already given in your lab manual, about which also you should be having an understanding, and please go according to the teacher who have taught you. Okay, general thing that I want to tell you. Also, 3 marks for preparation of this particular slide. Rest 2 marks is still remaining, so listen to me very carefully. First of all, let's see the general things which are there. Take onion bulb and place it on the test tube or glass bottle filled with water so that the base of the onion bulb touches the water. So basically over here they are telling you how to form the onion roots. So they are telling you either you can take the bulb and put it in a test tube containing water or put in a glass bottle containing water. The tip should be in touch with the water so that it will be growing up. Now after 5 days the roots start growing. Cut few root tips and transfer to a vial containing freshly prepared 1 part of glacial acetic acid and 3 parts of ethyl alcohol for 24 hours. Why are you treating it with glacial acetic acid and also ethyl alcohol, my dear children? Do you know that? Do you know? So glacial acetic acid makes sure that basically any sort of cell division would be stopped. Okay, all the cell division process which is there, it should be stopped, and that is why we use glacial acetic acid. Obviously there is more to it, but as of now this is a general level of understanding that at least you should be having. Now why do you use ethyl alcohol? So that the extra water content which is there in the cell, that can be removed. That is why you use ethyl alcohol. So both of them are mixed together and basically you put this root tips into that. Okay, this process is known as fixation, and then preserve the root tips in 70% alcohol so that no microbial growth and anything can take place. Now this thing you do not have to do. Okay, the people who are there in the laboratory, they might have already done, they might have already prepared the root tips as I have shown you in this particular video. So it was already prepared. What you have to do starts from this point. So while preparing a squash, take 2 to 3 onion root tips into a watch glass. Take it into a watch glass. Okay. Take 2 drops of 1 normal HCl and 3-4 drops of acetocarmine stain. So basically you are adding acetocarmine as well as HCl. What does the HCl do over here? HCl is also used for softening up the tissue so that it can take up the cells can take up the stain, which is your acetocarmine. Now you are going to gently warm the watch glass over the spirit lamp. Again, this is for softening. Okay, so that it can take up all the stain out there, and once the fumes start coming, close the watch glass with a petri dish. So make sure you do not have to wait for that generally. So basically after doing it, after heating it for like almost warming it for like 10 to 15 seconds, you can take the watch glass and basically cover it up with the petri plate. Okay, so that there is no drying up happening up there. Now after some time, the root tips, take the root tips onto a glass slide and then again add 1 to 2 drops of acetocarmine stain. Then you can place a cover slip and gently press the cover slip as well. Okay, with the back of the pencil, or you can also press with the help of your thumb finger. But you have to do it gently. Don't do it like this and break off the cover slip. It might be too much trouble for you. Okay, so the basic thing, as shown in the video also, acetocarmine plus HCl, you have to put it in the watch glass containing the root tip and then warm it up and then bring it up, cover it up with a petri dish, let it be there for some time. Then after some time, okay, after some time, take some root tip out there. If there is any extra thing out there, cut it off and then just put 1 to 2 drops of acetocarmine and then cover it with cover slip and then view it under the microscope. Okay, so this gives you 3 marks. Now what about those 2 marks out there? Those 2 marks you will get by identifying any one of the stage. So as of now, PU2 board has said that you do not need to identify any stages. But if at all in your school it's been like that, so please identify. But generally please ask, okay. Generally it is said that you do not have to identify, but at least basically writing about a stage would be really good. And also you have to comment on the stages absorbed. So basically any 2 points you have to explain over here. It will be really good if at all basically you explain, suppose you're taking anaphase, explain 2 points about anaphase, or metaphase and explain 2 points about metaphase, but please do that. You will get your 1 marks with that. Okay, so this is a general idea. If at all your teacher have told you something else to do and that's how you'll get your marks, please do that. Okay, so according to institution it might vary, but this was the general thing which was there. So previously you had to identify one stage and allegedly you had to comment on the stages observed and you have to write 2 points about it. But this time some things have changed up. Talk to your teacher also. Okay, so only the bottom line is that you have to be prepared with 2 points of each phases, know about it, okay, and please go and write it there. You will get your marks. Okay, and if you have any doubts regarding the 2 marks given over here, ask to your teacher at the earliest. Cool, so that works. I hope so. And also, if at all, okay, one more very important thing. The prophase, as I told you, it looks like this. Metaphase in the equatorial plane, anaphase, it is started separating, and telophase, my dear children, they are moved apart and they are at both the terminals out there. Cool. By the way, if you have not subscribed to the channel yet, subscribe to it. What are you waiting for? Subscribe to it so that you can get all the updates. And my dear children, did you like the video? Are you liking all the experiments which are being done? If yes, let us know. Okay, let us know through your comments, through your likes, through your shares. And also, if there is any doubt that you need to ask us, please ask us. So in the previous videos that I did, I had completed these experiments. So there was question number 1 where you had to pick one experiment and do it. It's not like you get to pick it. It's lottery system. Basically, with whatever chit you are picking up and whatever experiment you have got, you would have to do the experiment. So there was one experiment on pollen germination. I did that. There is pollen tube growth experiment. I did that. I also did the transverse section of ovary in hibiscus. I also did the nucleic acid staining. That was also done. So if at all you have not watched those videos, please watch because you will get 5 marks out of it. Now the next question which is there is this. Make the temporary slide of the given material, that is onion root tip. And over here you have to talk about the stages of mitosis. So this is the thing that I did right now. There are a few more things and that comes under spotting. It comes under spotting. So please stay tuned for it. The other videos will also be coming up. If at all you want anything specifically, you can let us know in the comment section below. Okay, so that's all for now. And also, if at all you're someone who is preparing for KCET, definitely check out the ABHYAS KCET test series, one of the most trusted test series. The previous year we got so many children who got great ranks. Obviously their hard work plus little bit of our hard work also. We had Prajwal with rank 171, Kiran with rank 443, and the list goes endless out there. Okay, so there are 35 mock tests over here and there will be chapter wise PYQs as well. And all of it in real exam like conditions. So 60 minutes, 60 questions, that's all. So test your speed and accuracy and you also get to understanding of how well are you performing in KCET. So please check it out. The link will be there in the description section below. Wishing you all the best and lots of love from Rasul ma'am. Please take care of yourselves and ask your doubts in the comment section. Bye-bye.